So I think I'm now online. Yeah, hello and welcome again to another Saturday live stream. Yeah, Microbehunter here, Oliver, that's my name. And yeah, over the next hour or so, I'd like to do again a little bit of microscopy with you. Like every week, a little bit of housekeeping here right at the beginning. Yeah, I think that the sound seems to be fine today. Yes, very nice. I see that people have already joined the chat as well. And what are we going to do today? Yeah, a whole bunch of miscellaneous material and stuff. Yes, I did announce that I would like to show you a Lego microscope. As a matter of fact, here it is. Okay, I'm going to explain it to you a little bit. You see that I've got a camera connected here as well. It's simply a little bit of a proof of concept, and you can also see the image that it's currently producing. Yeah, I'll spend a couple of minutes on that. I'll also put a couple of other objects under the microscope. I've got some hair from yeah, a dog here. I've got also some fur from a sheep here. Yeah, I'll be looking at some slides that I made several weeks ago to see how they essentially are right now. Yeah, and yeah, so a little bit of miscellaneous stuff. And I also prepared a few questions, not questions that yeah, that basically I came up with, but questions from my viewers that were posted on this YouTube channel. And I think I'm going to do this from now on as well. Every time that I'm going to always answer a couple of these questions as well. Please feel free to also ask questions in the chat. I will try to respond to them. Okay. So yes, for those of you who are new, please if you want to have if you have a specific question, then please say that at Microbehunter or at Oliver, so that it's easier for me to find to find the question in the chat. Okay. There's also the possibility between switching top chat and live chat. I'm going to think yeah. I'm going to do this here as well. Yeah, so basically, hello, I'm all around the world. What I'm going to do now is I'm going to quickly explain to you this Lego microscope and why I made it. The Lego microscope is actually I've made already several years ago. In my other channel, I also presented it. But essentially, the Lego microscope is nothing more than a Lego tube here. With a stage, and the light on the bottom, as you can see over here, is yeah, a little LED lamp which is battery operated. And you focus the specimen by pushing up and down on the stage a little bit. Now the image does not appear to be very much in contrast. Does not seem to have very high contrast. But actually, if you use eyepieces, then you're actually going to you have a very good image quality actually. Yeah. What I'm going to do now is I'm going to quickly show it to you. So this is the image that you see, but I also did prepare a slightly bigger view here. So what you see here on the bottom, that is of course the lamp, no condenser, nothing at all. And look, this one over here I made in such a way that you can move it up and down. The stage is basically contains a hole for the specimen over here. And look, I'm now going to move it up here. So there's a tube, of course, here. And here on the top, I added a camera. However, you can, of course, also add an eyepiece. And yeah, in here, yeah, don't drop it. And you can focus the microscope as well by pulling up and down on this plastic tube here. Now, this plastic tube, some of you might actually know what it is. It's an old film canister. These things can still be bought actually on eBay, but I think a cardboard tube would also do that, and I was just able to fit it in here. If you just look over here, yeah, see it's just square, and if you have the correct distance, then of course it'll work. The big question is, of course, now is what about the objective? And I would like to quickly tilt this over. I'm going to turn off the lamp as well. Yep. And if you look here, then here is the objective. It's a mirror image. Yeah, but how did I actually connect now the objective here? This here is a piece of plywood, just a piece of wood that I used. I used some hot glue to glue it to the Lego, but the objective is held in place over a little cap. Look at this here, what I got here. Yeah, this is put it down. That's how objectives are also sold, and yeah, when they're in here, then there is a little screw-on cap, and all I did is I drilled a hole here, and I also glued it on the piece of plywood, and that's basically it. And if the distance between the objective and the eyepiece, if the distance is correct, then you're going to get an image. It's really that simple. Yeah. So this Lego microscope is a little bit a proof of concept that it doesn't take a lot of effort to actually make something like this. But I mean, this here is a slide of a pine flower, and if you put it in here again, and if you switch over, where is it? Here it is. Okay, I'm going to add the lamp at the bottom, and of course with the LEDs that's not always very very centered, and then you can get yourself an image here. Okay, if the light comes a little bit from the side, then you're getting something a little bit like oblique illumination. But actually, the homemade microscopes actually work quite well. But it's not possible to make your own optics. I think if you want to have reasonable quality, you have to simply buy yourself an objective here. Yeah. Wouldn't you also get an image if the distance is shorter or longer? Of course, the magnification would be lower or higher. Well, the thing is the following. Yes, it is possible to, by changing the distance between the slide and the specimen, if you change the distance here, you can actually also move it further away. It's going to be difficult, and then the image is going to be a little bit blurry, but then you have to move the slide up to get the correct focus again. So it is possible indeed to change the magnification by changing the distances. I mean, ignore the fact that it goes out of focus, but if I push it upwards, then the image does become bigger. Okay, ignore the fact that it's out of focus now, right? You have to readjust the image, so it is possible indeed to do that. However, there is an optimum distance between the slide and the objective, and if you do not keep that distance, then you're introducing other problems, like, for example, it's not going to be clear on the edges and so on. Okay, so yes, it is correct. It is possible to also change the magnification by changing the distance, but this only goes up to a certain point. Okay, so I've always wondered what controls the behavior of the microscopic world. What does paramecium look like under? Now, I do not have. Well, I mean, I do have. Your request. I will try this now. I mean, I don't have paramecium here, but these are other ciliates. And there are a couple of other ciliates on here. Now, these are probably pretty difficult to observe because they don't have a color. But you know what I'm going to do now is I'm going to just show you a little bit here. Yeah, now it's a little bit smaller. Maybe I can change the, let's do it like this here. Okay. So what I'm going to do now is I'm going to move the Lego microscope a little bit out of the way. What I've got here is I have some pond water, and I added a little wheat grain, and I left it basically rot for a week or so. And there are a few ciliates in there indeed, but not paramecium. Unfortunately not, but smaller ones. So this is they're gonna be a little bit difficult to see. And what we can do now is we can actually compare now how this looks like under the Lego microscope compared to my good microscope. Okay. I'm slowly running out of cover glasses as well. Okay. So let's simply drop it on here first, and let's give it a try. Always remove the water sample again because I don't want it to tip over. And this is probably not going to be easy, okay? Because they do not have a lot of contrast. Yeah. So, and for this reason, but maybe that's still a good example because it kind of shows maybe the limitations of everything. I mean, lots of stuff floating around, but I don't see any movement. Where are they? Maybe I got it from an unfortunate place. There's lots of debris. I mean, today in the afternoon I got it much better. Okay. You see the contrast is not particularly good. So what I'm going to do now is I'm going to switch over. Ah, I saw something. There was one. Oh, there's another one here. Oh, there are a couple here. Okay, so I think it's yeah, just a few days ago the concentration of microorganisms was much higher. Yeah. Up, and now I bumped against it. That's always, of course, a problem. Yeah. Lots of bacteria are in here. Yeah. Yeah. But yeah, you see that I'm moving the lamp now a little bit. Yeah, so this is basically how it looks like. Again, there is a somewhat of an internal in the tube, oblique. Look, this is actually quite nice. When the lamp is from the side a little bit, then yeah, it looks a little bit 3D. So that's a form of oblique illumination as well. Okay, and kind of also illustrates that yeah, you can also get different lighting techniques, even though it does not have a condenser. So the image quality would of course be much higher with the presence of a condenser, but I think yeah. So this is yeah. How do you recognize that they are bacteria? Bacteria have a specific essentially size and also shape. So maybe I'm going to show you over here. I'm going to turn on the arrow over here. How can I arrow? Here I cannot move the arrow. Do you see this long thing here? Yeah, that's almost certainly bacteria. Yeah? So very characteristic. Yeah, but the contrast is not very good. But still, it kind of shows that it is possible to see bacteria even with this homemade microscope. And even though it does not have a condenser, by the way, this has now a 10 times magnifying objective. So, but I know that you probably want to see how this actually looks like with a condenser present and with proper optics and everything. Okay. So what magnification is the question here? I'm using now my 10 times objective, and concerning the magnification, lots of people want to know what is the magnification, and I'm going to tell you somewhat provocatively. But actually it's not wrong. The magnification depends on the size of your monitor or on the size of your mobile phone screen. Yeah, now some of you might be a little bit surprised, right? Because the larger your screen or the larger the video that you're looking at, the larger the magnification. Because magnification is the size of the image that you see on the screen divided by the original size. So if you ask me what magnification is it, then I wouldn't be able to tell you because it depends on your monitor. Now, this is not a satisfactory answer, and maybe not an answer that you expected. Yeah, because indeed there is also magnification, of course, written over here on the objective, right? But this type, like for example, in this case, it's a 10 times objective. But this actually says nothing about the total magnification. Yeah. So there are actually, the word magnification can mean different things here, okay? So what I'm going to do now is I'm going to switch microscopes, and we're now going, as a reference, we're now going to have a look, yeah. Oh, I have to turn on the other microscope first, of course. Okay, so that's the one that you're now seeing here on top. And I'm going to put the slide in there. I'm going to save the battery. And if you want to, in a couple of minutes, I'm going to take the microscope, the Lego microscope apart to give you a little more details. Yeah, so. And let me see, let me turn up. Let me turn this down. So this is now the 4 times. So this is now the same magnification. And you're seeing beautiful blue colors here. Okay, why is that? Well, it is because of my polarization setup that I have. It's called DIC. And you're going to see over here a little bit. Where is the arrow? Did you just see those microbes before? And now they are here. And here it looks actually much smaller. They look much smaller, actually much more difficult to see. Now, how is that? It's the same, the objective has the same magnification. Well, because this depends also on the field of view, because essentially my camera, the one over here, right, magnifies more than the camera that I have on the other microscope. So you see that magnification depends on a variety of factors. Yeah. And I'm going to now move up with the magnification, change the filter around. Yeah. Yeah, and now you can actually see that the bacteria here are in much better contrast, but all of the other ciliates that were moving around just before, I have a problem finding them, because sometimes what happens is that they move to the side of the cover glass. Here, for example, because at the side of the cover glass you have a higher oxygen concentration. And all these things here, those lines here, these are all bacteria, almost certainly. Yeah. So, and this is actually a sign, if there are many bacteria present, then this is a sign that ciliates like Paramecia and so on are not going to be present so much, because they consume so much oxygen that essentially it's not, This is also a sample where I would be careful a little bit. You don't want to mess around with this sample, because you don't know which bacteria these are. It could be, you do not have to assume that they're automatically dangerous, but actually you don't know what you're growing. Yeah, it's the same time when you decompose some food. Yeah, you don't want to know, you don't want to eat it. Yeah, or touch it. Yeah, so so this is now at 20 times magnification, and let's go up further. Okay, so I just wanted to show this to you a little bit as a reference to the Lego microscope. Yeah. So I'm going to quickly have a look at a couple of comments here. I'm listening. I'm listening my first live with Oliver, and I received 10 minutes ago my camera and 200 prepared slides from a new microscope. Okay, yeah. I think it's called display magnification. It's also used by many manufacturers of digital microscopes, and it's how they come up with the ridiculous magnifications of 2,000 times, etc. This could be one thing. However, when the people say that they magnify 2,000 times, do I have this here now? Then this is sometimes because what they do is they use, here's one, they use sometimes eyepieces that magnify, for example, this one only magnifies 15 times, but there are eyepieces magnified 25 times, and this way you can artificially blow up the magnification. And it's just a marketing thing. Yeah. Is it safe to keep that in your room? Let's put it this way. Every time when you're dealing with unknown bacteria, you have to have a healthy respect. So if you look at this jar over here, where I'm going to switch over now again. I'm going to switch away from I'm going to show this to you. So let's put the microscope away. So what I've done is, this is called an enrichment culture. And you see it looks pretty yucky, okay? It's slimy and so on. So generally, what I've done is added a little of pond water, and I added a wheat grain. Let me see if I've got some more left for demonstration purposes. Just a second. I think I used them up today, or I don't know where I put them. Here. Okay, here it is. Here's this. This here is a wheat grain, whole grain. The small dots that you see here are this yeast. And what you do is you take a wheat grain, which contains, of course, a lot of starch. You crush it between 2 spoons, and you add it to the pond water, and then you wait a week. And it's going to turn pretty nasty. And there are, a lot of bacteria in here, and you don't know essentially which ones. So you need a healthy respect. So I would not touch this with my fingers. I wouldn't automatically assume that they are dangerous or so, but the concentration of bacteria is quite high. It's basically the same. If you ask me, is it dangerous to keep it in your room? Is it a little bit like the same as asking how dangerous is it if you keep food in your room which starts to decompose and rot? It's also not healthy. Okay, so I would actually consider this in a similar level. Yeah. So this is, should I use the camera that came with my microscope to see better? I tell you the following. It's a question of taste. If you want to have a camera connected to your microscope, then it can be more convenient if you have a screen, for example. And some people who have some vision problems might actually like to work more with a camera and a screen. But however, the image quality is much better generally if you look directly through the eyepieces. Okay, so this is a little bit the thing. What I'm going to do is I'm going to now, flip around the image, and I'm going to just show you the Lego microscope in a little more detail. Again, the purpose here is I'm going to unplug the thing now. The purpose is a little bit to give you a demonstration here. Long time ago, 20 years ago, 30 years ago, I still am interested, but I used to be quite interested in astronomy, and I even attempted to make my own telescope. So hobby telescope making is a thing that many people do. However, hobby microscope making is not so common, okay? Also because you cannot make your own optics. Yeah. So I'm just going to show this to you. This is how it looks like, and this is essentially all it is. It's just a tube, and here on top, it's just a tube. And I'm going to now. This is kind of the main part down here. You see that I glued the ring from a container. From a container like this for storage of objectives. I glued it on here. I drilled a hole, and then I also used glue. And can I even remove this? I have to be careful here. Okay, here we go. Okay, this is how it looks like. So there is some plywood here, and I used some hot glue to, let me do it like this, to connect the first layer of Lego pieces. I know that some folks have 3D printed certain things here, and it's not the first time that somebody's making a microscope out of Lego, but it's simply all you gotta do is you can cut it also with plywood. I'm just saying the following: if you wanted to make something like this, those objectives can be bought fairly cheaply. Okay, I do not recommend too high magnifications. 10 times is fine. You need a 10 times eyepiece as well, and what you have to make sure is that there is approximately a distance of 160 millimeters. It says here over here 160. Okay, it says it's flipped. Okay, mirrored. Unfortunately, it says here 160, and this 160 millimeters is the distance between the top part of the. Let me unscrew this here. The top part of the microscope objective to the eyepiece. So that's kind of the standard, and if you kind of keep this distance properly, then essentially you have a microscope. That's all there is to it. So this is a little bit for those of you who are interested, it's a little bit of a weekend project. Okay, so I just wanted to show this to you. Now I took it apart. I think I'm just going to leave it apart now, and I'm just going to put it away because I would like to show you a few other things here as well. Okay, so I'm just going through the questions again. Can be varied with the camera, such as zoom capability. Yes. We are constantly in contact with bacteria, and people even go swimming in ponds and lakes. What makes the bacteria in your jar more dangerous potentially than the ones around us? Good point. Good point. And I will tell you the following. They're not necessarily more dangerous, but there's one difference. There's one important difference. So your point is completely valid. We're always in contact with bacteria, and as a matter of fact, there are more bacteria growing on your skin and in our body than we have body cells. Can you believe this? There are more cells on our body than we actually have body cells. So, but your question is now, why should I be more concerned with those in here than with the bacteria that we are in constant exposure? And the difference is that the concentration here is significantly higher. That's the thing, okay? The concentration of the amount of bacteria is significantly higher here. And that's it. And if there is a small wound, for example, a small cut in your finger, and some of them are, I don't know what they are, I do not have to assume that they're harmful, right? But essentially then the amount of bacteria that will enter our body is significantly higher, and the immune system might not be able to handle it. That's it, yeah? So, however, I also have to say the following. This here is a pond water sample. And I would be more concerned, for example, if I were to decompose meat, okay? So for example, if I take some meat and I were to add some, then this, I would be a little bit more concerned, because those bacteria actually have the possibility to live on meat, on animals, right? And humans are animals, right? But these are, under quotation marks, only water bacteria. Yeah. So I'm not saying, I'm just still saying you got to be reasonably respectful. Yeah. As a matter of fact, if you want to grow unknown bacteria at home on a petri dish, I do not recommend that. Right. You're automatically in an elevated biohazard category number 2. So that's something that actually only authorized labs are allowed to do. Yeah. But then, by the way, in an upcoming stream, I want to actually show you how I'm growing E. coli bacteria. Yeah. So. Can we see bacteria under dark field of microscope? Is yes. How much magnification is required to see it? The following. Look, I only have dark field for the low power 4 times objective. Okay. And I'm going to pull this out. And I have to open up this here. Okay, that's the low power. Okay, 4 times. Am I able to see the bacteria? These are the tiny little dots. The answer is yes. Even at 4 times, right? But the important thing here is that they're so small that I'm not able to see the shape, right? So this is a little bit of a limitation of my microscope here, because actually this is not a dark field filter, but it's actually for phase contrast. Okay, the phase contrast patch stop, but which also works as dark field. And look, if I change the condenser around, that's a little too much. Yeah? So the short answer is, are you able to see bacteria with dark field? The answer is clearly yes. Okay. Let's have a look now at phase contrast, just because some of you might want to be interested. And this is now phase contrast, and look, and this is of course, a typical phase contrast application, because now the bacteria look very dark. Okay, look at even some here in short chains, and the phase contrast optics convert differences in refractive index into brightness differences. Yeah, some of them are even moving around. Okay, so I want to show you something else here. Okay, something that is also prepared. Bacteria is one thing. Again, I think bacteria are not the most exciting specimens to look at, but people are interested in bacteria. I get it. So what I'm going to do now is this slide here. Okay. Containing the bacteria, I'm just a little extra, will receive a separate treatment. Yeah, so I'm going to add a little bit of alcohol on top of it later to disinfect it. Yeah, so but I want to show you something else. Just a second. I need to reach over into my cupboard. Here we go. Look what I got here. Let's have a desk view again. This is for people who have an aquarium. Probiotics, it's called. Increases disease resistance, okay, and inhibits the growth of harmful bacteria, increases survivability and stimulates digestion of what? Of fish. Okay, these are freeze-dried bacteria, which you add to an aquarium. Doesn't smell good, right? And basically these are bacterial cultures that pure bacteria. What you see here are pure bacteria, and what you do is you can add them to an aquarium and then start reproducing. And what I have done is I've taken a few of those and dissolved them in water. Okay, because some people want to see bacteria, but not dangerous ones. Yeah. So yeah, I've put them in here, and I'm going to take a small sample, and put it on here as well. Yeah, today, as you see, I'm doing a little bit of random stuff. Lego microscope, observing bacteria. I was actually hoping to get some Sahara sand from the air, and which turned out not to be Sahara sand. That's another thing I would like to show you. So, and I have to find, as always, the thing here. Okay. And let's put those probiotic commercial bacteria under the microscope. Okay. Here we need to go up with the magnification a little bit. Those clumps are way too large for bacteria. Okay, so this 40 times. Okay, it's a little too dark. But those, I know this might look a little disappointing now, okay? But those tiny dots that you see here are bacteria, okay? Very low in contrast here. Yeah. So, and in phase contrast, remember the one that increases the contrast a little bit? I have to open it. I forgot to open it. Yeah, you see those tiny dark dots here. These are the bacteria. Okay. Yeah, they're not moving around because they need some time to regenerate because they were just freeze-dried. What is this? What? Why is there this thing moving around? Okay, this must be clearly a contaminant. Okay, that's way too large for bacterium. And I don't know, it must have somehow found its way into this tiny little dish here. Maybe I always exchanged the pipette tip, so it couldn't be a cross-contamination. That's interesting. Okay. Yeah, but you see, I just wanted to show this to you here. Bacteria are small and difficult to see. You see over here, some of them are even in tiny chains over here. Yeah. So I'm going to quickly read again. If you put probiotic bacteria in there, would they eat the pond bacteria? Probably not. Bacteria eating bacteria, I mean, yeah, I think that's the way it works. It's usually like this. If you have the so-called the good, under quotation marks, bacteria around there, what they're going to do is they're going to rapidly multiply, and the so-called the bad bacteria won't have enough resources to multiply as quickly. Okay, so that's a little bit the reason, right? You overgrow the bad bacteria by adding good bacteria. Yeah, so that's yeah. Do you have an amoeba or euglena sample? I mean, not euglena, but I do have some pond water over there. But finding an amoeba in there is a little bit a question of luck. Okay, so yeah. So the answer would be, yeah, I could find some maybe, but it's gonna probably take 20 minutes or so until I'm able to find an amoeba. Okay. So I hope I didn't forget any questions here, but otherwise I'm going to move on a little bit. Yeah, so these were the bacteria. What else did I want to do? Okay, what I'm going to do now is I'm just always checking the time a little bit. I'm going to, for a couple of minutes, answer 5 questions. Okay, I'm going to do, I decided maybe to do a little theory part for a couple of minutes in each live stream. And then later on, I want to put this sample under the microscope here, which is not Sahara sand. I'm going to tell you why I was hoping for Sahara sand. So, and yeah, this is a question that I got. So I just simply copy-pasted it as it is, so forget about the English here. A snail could be seen with a naked eye. So basically I put a snail under the microscope in one of my videos, and it could be seen with a... Why do you actually need a microscope if you can see the things also without a microscope? This is kind of the question here. Okay, it was when I read it the first time. This question the first time, I was confused. What am I supposed to say here? Okay, if you can see, the snail was a tiny snail without a microscope. So why do you even need a microscope? And the answer is, of course, is if you look at things under the microscope, you see them much better. Where did I get the snail from? In one of the videos that I just released yesterday, I made a mini aquarium for water fleas, and there were some snails in there as well. And yeah, this is, no, that's not the one. I have to, where is the camera? Ah, okay. Let me see, I have to reattach the camera. I hope that I'm able to make this to work. Just a second, I have to change the camera. Then I have to, let me try this again. I hope that I'm able to reactivate the camera. Ah, yes, we're lucky. So this is now my stereo microscope. Okay. And this is, look what I found here. Okay, it's a little out of focus. So under the stereo microscope, yeah, so the snails are around 3 millimeters in size. The camera always adjusts the exposure time. But I mean, this is the reason why we put them under microscopes, because they look much better. Yeah, and you see details that you would otherwise not be able to see. So this is basically, see, the camera always adjusts its exposure time, right? Yeah, so, and that's essentially what I like doing with hobby microscopy. Sometimes I just basically, it's not complicated science that I'm doing. It's just I'm having a little bit of fun observing nature, because by observing nature, we're also expanding our horizons. So let me quickly make it a little bit. Here we go again. So the bottom line is, while we are able to see, of course, many things without a microscope, obviously, with a microscope, things sometimes simply look much more interesting. Okay, you get the idea. Yeah. So this was essentially the next question. Some of them are also quite advanced. Talking about water fleas, the water crustaceans like Daphnia, Cyclops, or the ostracods, and so on. Yeah, I basically simply refer to them as water fleas, as a collective term. These are tiny water crustaceans. Fleas are actually insects. Their insects actually are not really closely related to them. They're both arthropods, but yeah, still. And the question is now the following. Let me go back here. Yeah. Water fleas, can the water fleas live inside cats and make them sick? And again, here it took me a couple of minutes to actually understand what the question actually means, and then I realized that cats and dogs can have fleas, right? These are the blood-sucking little insects, and now I put water fleas under the microscope. So I think there was people assumed that there's this kind of connection, and this shows a little bit a problem in biology, that sometimes you use terms for things that are essentially not related, right? Water fleas and fleas that you find on the blood-sucking fleas are not, they have nothing to do with each other. Yeah, one of them are insects, the other ones are crustaceans, belong to the crabs. Yeah, so this kind of illustrates a little bit that in biology and in other areas, naming, sometimes the naming can cause some kind of confusion here. Okay, so and I do have. This is here's another question over here. Magnification, and this is essentially was a request beneath one of my videos here, and I already talked about this here because a similar question appeared here. What is the magnification? And you might have noticed that I do not put any magnification into the corner, for example, and I also do not put a scale bar. And the question is why not? The reason is which magnification should I put on here? If I say, okay, 10 times, then what does this mean? Does this mean that I'm using the 10 times objective? Does it mean that it's magnified 10 times? Actually, it's got to be more than that, right? So this is a little bit confusing because the word magnification can mean different things. And even if I put or as I explained before, if I use a different definition of magnification, then the actual magnification would depend on the size of your screen, Right. So this basically is causing a little bit of a dilemma in me, because whatever number I put on there, it's probably not going to be correct, right? And it's going to be confusing. And this is the reason why I do not include the magnification. And also, what happens if I use a certain objective? Let's I'm going to show it to you again. Okay, here let's say I'm using the a 40 times objective. I'm going to give you a more interesting slide now. Okay. Where, yeah, that's the one from the pine leaf, for example. Yeah, I'm going. Let's just have a look at this one here. Okay. Okay, so this is and I'm right now using the 10 times objective, but actually on screen it's magnified more than 10 times. Yeah. And what happens now? Just to illustrate this a little bit. I'm just going to illustrate this now. Just a second. Look what I'm doing now. I'm not changing the magnification, right? So what am I supposed to write now on the image? Yeah? So this is a little bit a thing that is a little bit difficult for me to do. Of course, I can add scale bars. This would of course also work, and then you'd have a scale bar from going from one place to another, and it says 10 micrometers. Right? Yeah, but how much is that? So it's also not always very useful. And for this reason, I have not yet found a satisfying solution here concerning the magnification issues. Yeah. So I'm going to go through here. I hope I got the sequence correct. Dark field. Do you have amoeba? Okay. Some people are saying hello. Would I need a big microscope to see white blood cells? What is a big microscope? So that is the thing. You can see again here white blood cells also at a relatively low magnification. Okay. But maybe not very well. So it's a question of how much magnification do you want to have? And generally, even small microscopes will magnify. Maximum magnification, total magnification is 1,000 times. Oops. I think my video froze. Could this be the case? Let me just see. Now my face is gone. I hope that I'm going to reappear. Uh-oh. What happened now? But I think you should be able to hear me. But somehow my face is gone. This is a little bit unfortunate. Okay. Audio okay, but video is gone. Yeah, I know. I can see that there is the audio does work. I can see this indicator, but for whatever reason, this is pretty frustrating. And if I restart the OBS Studio, then essentially, the live stream stops. Can we see an opaque object with a compound microscope? Because I'm interested in collecting micrometeorites. Yes, you can do that with a low magnification by using a strong flashlight from the top. But I'm a little bit frustrated now myself because I cannot make the thing reappear. There is how do you call this? An emergency solution. Look what I'm doing right now. I think this is gonna be. Is this gonna be the? I guess this is gonna be the emergency solution for right now. Look what I'm trying to do now. I'm going to try to maybe remove this here. I can try to then now use this here, and maybe I'm able to make this a bit larger. Can I do that? It's not ideal what I'm doing right now, but maybe I can move this a bit over. But I think it's better like this than nothing at all. I hope that's going to be fine. But this basically means that I have to, just a second, give me a minute. I have to copy now this video over also to the other scenes. This is not fun. And the desk view over here. And here we go. Okay, I guess we got to do it like this. I hope you don't mind. Okay. So unplug the camera and plug it in again or remove it. Yeah, so that is something that I also try to do sometimes, but then it might not find it again. You know what I'm gonna do is I'm going to try it in any case, because at least we got this one here working. And if the other camera doesn't work, then yeah, it's for whatever reason. You see, just a second. Yeah, see, it's not visible. I'm going to try to unplug it. Just a second. Unplugged, and I'm plugging it back again. I hope that it's able to recognize. Still not working. I think I have some kind of an. Sometimes the computer is a little bit, yeah, over. How do you call this? Exerted because I've got too many cameras connected. This is a little bit frustrating. So what I'm going to do now is I'm going to try to deactivate it, and I'm going to. Nah, you know what? I'm just going to continue like this. It's yeah, better than nothing. So. I'll simply move on here. So this one is also an interesting one. This is just a second. Can I make this a little bit? I'm going to remove the background here, and I'm going to remove this here. So, can you recommend some videos or readings on how to test seawater for dangerous levels of E. coli bacteria and feces pollution due to sewage spillage? I want to start monitoring nearby beaches for a study, but would love some advice on what equipment I would need to and some guidance. Thanks in advance. So first of all, not with microscopes. Okay, you cannot do serious identification using microscopes. The reason is that microscopy, the shape of bacteria, does not tell you anything about what they are. It's a little bit like trying to identify the make of a car or the brand of a car by looking at its color. It's not going to work, right? Because whether bacteria are dangerous or not, or what they are, depends on their DNA, which you cannot see obviously, and or the chemicals that or the toxins that they produce, which you cannot see under the microscope. So you have to use biochemical methods or culturing methods and other identification methods, but microscopy unfortunately is not going to work. However, for laboratories, there are, of course, certain test kits available, and you can also, of course, grow them in culture, and then you can have so-called elective and selective growth media. It's all pretty fancy, but I would say that if for serious things like health issues, that's not something that you just can do yourself. Yeah, I wouldn't risk that. Yeah. So for magnification, maybe you could now and then again mention that you can easily see this or that under the 20 times or 40 times on the microscope. That way we will know what to expect. I think this is actually a good point. I was actually wondering myself a little bit, why do people always want to know the magnification? I mean, because, yeah, but maybe I've been overthinking this a little bit too much. It could indeed be that people want to know, am I able to see the same thing with my own microscope? Yeah. So what I've done is, therefore, in the past once, and I might think about reintroducing this by indicating the objective that I've used. So, for example, I used the 10 times or 20 times objective. Having said that, whether it looks similar under your microscope depends, of course, then also on the field of view of your camera, right? So this, is also something that has to be included here. Yeah. To compare size, I always have two solids next to each other. One with a human hair so that I can have an immediate comparison. Yeah, that's also possible. Yeah, to have some kind of a reference. Yeah. Another reason I enjoy love your videos, all the mistakes and problems. That's real life microscopy. Yes, that is indeed a little bit this authentic nature as well. It's as a matter of fact about videos, I did receive some feedback also from other people that says don't edit the mistakes away, right? It's kind of keeps the authentic nature. Yeah. So, do you think there is a way to use, and that's the last question here, to use some kind of special container similar in volume to yours to observe microorganisms, or without collecting them from the container, or two vessels connected by thin tunnel, like two glass slides, or viewing with a regular microscope? Okay, I'm going to show you something. Again, I have to reach down into my drawer. I have the following here. I used once or twice already. It's pretty delicate. It's called a Microlife, they call it. Okay, I just want to show this to you. It's a pretty delicate device. And look, there it even comes with a pipette here. And indeed, there are 2 containers here, right? And there is a small tunnel, or I don't know, what do you call this, path here. So what you do is you can actually fill in a water sample in here. It will spread, and it's thin enough that actually the microorganisms can actually move along. And then what you can do as well is you can actually use a pipette, and you can actually, you see there's a rubber, cone here, and then you can actually also push the liquid through the whole thing here. Also for cleaning. So yes, this does exist. Disadvantage, I think, of this is it is kind of delicate, and I wonder if it is not somehow possible to make something similar yourself. Okay, maybe using some kind of a large cover glass or so. I don't know. But indeed, this does exist. I think there's something else in here, right? Here's it. Here, that's this one over here, and including of course an instruction. So this is what I've once done, and it was several months ago already. I put a culture with paramecia in here, and then I added an electrical current. So, and you could actually see that the paramecia, they are following the electrical current. You could actually move them, go left and right, depending on the polarity of the electricity. So this is actually quite useful. However, I did find it also a little bit difficult to clean. You have to rinse it out, but then of course sometimes some of the microorganisms or so might actually get attached in there. So this is essentially you were directly your suggestion over here was actually directly is already solved, so to say. It does exist. So, okay, let's I'll have another look here. Why or how it works on the agar that the cultures have colors. For example, total count is red on the test we use in our lab. Okay. So, this can, again, your question can also be interpreted in different ways. I don't know which color you are now referring to. Are you referring to the color of the colony, or are you referring to the color of the agar? If you're referring to the color of the agar plates, the culture plates, then some of them are red because it's called blood agar. So there is actually sheep blood in it for certain bacteria that need blood. So these are sometimes hemolytic bacteria. They feed on blood, and that's why their agar is red. Because there is actually blood in the agar medium, in the growth medium, in the petri dish. Okay. If you're now referring to the color of the colony, then this can be due to 2 reasons. First of all, some bacteria do have their own pigments. So that's number 1. And sometimes it is like this, that there is a so-called an indicator substance inside the agar. And if the colony grows, then it changes the pH. Very often bacteria produce an acid. And this acid will then react with the indicator and will make it red. So there can be a variety of different reasons. I'd have to know a little bit more about this. But this was already quite technical here. Okay, so what I'm going to do now is, what time is it? Okay, it's almost an hour. I'm going to move on a little bit. I want to show you a few other things here. Okay, as promised, today's live stream is a little bit, I would say not chaotic, but at least a little bit, has a variety of topics. And I would like to show you now a short video that I made here. See, this is again something where you do not see me. So what I'm going to do again is I'm going to copy myself over to the video over here so that you're also able to see me. Look, this is on my car window about 2 or 3 days ago. we had Sahara dust in the air. At least this is what was announced yeah, over the news. Every now and then, if the weather is right and the wind is right, and there's a sandstorm in Africa, then sometimes the Sahara sand is actually carried also into Europe, where I'm living. And I was so happy. Wow, we've got some Sahara dust finally, right? And even if you look on the horizon, you could see it was slightly yellowish, yeah. So there was quite a bit of haze around, and then I found all of these things and collected them. Here is the tube, and I said, "Cool, I'm going to put some Sahara sand under the microscope." Big disappointment. It's not Sahara sand. You might have already guessed what it is. Yeah, it's pollen time. Yeah. So unfortunately no Sahara sand, okay, and I said, okay, yeah, I'm still going to put it under the microscope now, and yeah, so here we are. Okay, now let me put this over here. I've got this other camera. Yeah. So this is actually pretty straightforward again. I'm going to not make a permanent slide. I'm going to just take a little bit of water. You cannot see it. I've got a glass of water behind here, and yeah, here a small drop of water. I need some tissue paper for the remaining water. Yeah, and I think I'll take this mishap with a frozen video as a, how do you say, an excuse to buy myself some stronger USB cards for the computer, because I think that the USB is a little bit overloaded, because I've got, I don't know, I've got how many cameras do I have connected? One on my stereo microscope, then the one here, the desk, 2, 3, the one that I'm looking in right now for the stage, 4, the other camera on the other one over here, a microphone, lots of cameras. Huh? And I think that every now and then this could also be an electricity problem, that essentially sometimes the cameras stop working. Yeah, so I picked up a little bit of, simply went in here, picked up a little bit of this, what I hoped to be Sahara sand. Yeah. And which it was not. Yeah. But that's also sometimes interesting to see that. Yeah. And then I lost my cover glasses. And here we go. Just going to drop this on here. Up. And now we have another look at, no, not this one here. No, that's the one. That's the correct one. Okay, let's remove this. And that's what I got. Okay. Yeah, pollen, not sand, or at least mostly pollen. Huh? So this is, and a whole bunch of air bubbles right now. So that's basically, the Sahara sand, which wasn't Sahara sand, at least. Yeah. So, but still looks kind of nice. Yeah. So, this is, yeah. So let me go through the comments here again. I'm reading through the comments. Lichens and moss from trees works well for tardigrades. Yes, I can talk you a little bit here, the following. Maybe I'm going to do in the future again another tardigrade session. If you want to collect tardigrades from moss, just make sure that at least it's my experience, there are many places where moss grows, but not all moss is equally suitable. So my suggestion is that you choose some moss that has been indeed in a forest. I've been much more successful. Even if the moss is dry, if you add water, you're going to find way more tardigrades than moss that is growing exposed on a rock somewhere. And I was kind of wondering why this is the case, and I think the reason could be is that exposed moss is exposed also when the sun is shining. It dries up completely and is exposed to more heat. Now tardigrades apparently are quite resilient for drying, but they don't like heat. And if it's too hot, then actually they don't like that. So if you want to collect tardigrades, get the moss from a place where there is, where it's basically from a forest. Yeah. Those pollen are split in two. Does this mean they're sprouted? No, I don't. No. Because that's also a good point here. No, that is the way that they actually look like. Because pollen will not sprout like this. They will form a pollen tube. In that sense, pollen are different than seeds. I think that's where the association comes from. I just show it to you again here. Yeah. So if pollen, when they actually start to germinate, what will happen is they will start to form a tube growing out. And then in this tube, this tube will grow. I'm looking into the wrong camera. I just realized. And this tube will then actually contain the sperm cells that will actually then fertilize. Okay, so this is not because they have started to sprout, but that's actually their shape. Yeah. So. Have you looked at the micrometeorites suggested by another participant? Yes, I have. Indeed. However, this is another thing here. You cannot be absolutely sure that that these are micrometeorites. You'd have to do some additional testing. Just short clarification. If you take a strong magnet and if you go over the surface of, let's say, where rainwater is collected, and if you collect all of the magnetic material with a magnet, and if you put it under a microscope, then sometimes, and I've also found several of those, you're able to see very round structures, extremely round. And these could be micrometeorites, cosmic dust. And essentially when it falls through the atmosphere, they start to melt and form those perfectly round spheres, microscopic spheres, and then basically you can collect them with a magnet. However, many of those can also be of origin from the Earth. So for example, if there's a company somewhere, and it's kind of welding something, and then those things can also appear to be similar. Right. So in order to really determine whether these are micrometeorites, you actually have to do additional testing. Yeah, maybe you have to analyze the crystal structure or the isotopes or whatever. Yeah, but still, independent of that, of whether it's the origin from Earth or indeed from space, they're kind of fun to observe. You know what? Yeah, I'll take this as a suggestion, and maybe in one future live stream, I'm also going to show you some of those. Yeah, but indeed, there are also several web pages available where they talk about micrometeorites. But to be really sure that they are micrometeorites, you'd have to do additional testing. Yeah, because they could be origin from somewhere else, from Earth. Okay, so then what I'm going to do now is just a little bit for fun. Because this has a little bit of a tradition already, even though I already know what to expect. This is because it also looks kind of, you know what this is, right? Yeah, dandelion seed, and just for the fun of it, let's have a look. I mean, the snails. This one snail is actually already out, has escaped. So what I need to do is I'm going to pick up the snail. Let me quickly switch over to the here. That's one. See this also. This one also has frozen. It's also kind of weird. So let me quickly, just a second, stay. See, this is there seems to be an issue here. Yeah, it seems to be this one over here also has frozen, but maybe I'm going to deactivate it and I'm going to activate it again. Yes, now it works. Okay, so here's one snail. The other snail is over here on the side. Yeah, here it is. I need to pick it up somehow, and I'm gonna dump it. A little piece of paper. A piece of paper might do the trick. Here it is on a piece of paper. Okay, let's put it back into the. Here we go. It's back, and I wanted to put some of these things here under the microscope. Stereo is also nice sometimes. Here it is. Ooh, way too bright. You just are able to find it, but you know what? I'm wondering if maybe I should not turn on the light from the bottom. the wrong lamp. Here we go. Where is this? I don't know. Yeah, but in any case, yeah. So that's basically how it looks like under the stereo microscope. Not particularly good. I just realized I have to optimize this a little bit. But yeah, the shoot here of the dandelion seed actually looks quite good also under the compound microscope. But you have to, of course, remove the seed first. Okay, and this is actually also a little bit of a nice test to see and which mounting medium actually works best. Yeah, so I put it on here. I don't remember if, I just dropped my pipette, and a little bit of water again. And I'm going to put some water on here. Cover glass goes on top. And I would like to add a little more water here on the side. Okay. And let's have a look. I need to... So, let's go down with the magnification and see it does not fill the full field of view. Okay. So, not particularly. You have to swing out the condenser. So this is how it looks like, okay? And what we notice is, of course, that those tiny little fibers have a certain structure to them. Okay. And turn up the whole thing here a little bit. And indeed, sometimes people will find those fibers also in water samples and in nature, and then they're wondering what could it be. And especially those, yeah, those tiny little spikes here. So I've seen those fibers, found them fairly often also in dust already, also in some water samples. Yeah, and I just wanted to show you that if you find those, it could indeed be the shoot of a dandelion. Okay, so this is the thing. Yeah. So, some people are talking about cameras right now. Okay. I wonder how the tardigrades on the moon are doing. Ooh, see, this was actually some water spreading. Okay. Yeah, they actually, some time ago, they sent some tardigrades to the moon as well. Yeah. The snail poop on the slide might get lots of digested algae. There was a little bit of snail poop indeed somewhere. Yeah. I can actually maybe, I'm going to put it, here. A microscope camera into an iPad. I think there must be certain adapters. I know I used to have an iPad many years ago, and So, do you sometimes see your own fingerprint on the slide? The answer is yes and no. I will tell you why. Because if you have a fingerprint on the slide, sometimes it is so big that you do not recognize it as a fingerprint. And generally what I do is before I use the microscope slides, then I will usually dry wipe them. Yeah, to remove any grease or oil or fingerprint. Yeah, but if there is indeed a fingerprint on here, then usually it's not only a fingerprint, but there's usually some other dirt and dust as well, which might be a little bit disturbing. Yeah. So so HDMI. Yeah, so some people are talking about connecting a camera. If you want to know what I've got is the microscope. You know what? I'm gonna just So that is my microscope camera that I have, right? It's a Canon D90, and it's a 4K camera actually, and I also connected it over HDMI over an HDMI to USB converter, and then I basically put it into the computer. So that's essentially how you're getting the picture right now here. Yeah. So that is just something I wanted to show you, and you can see that it's sometimes those fibers are moving a little bit because they're absorbing the water. Okay. So, and two more things that I would like to show you, and these things are, the hair, and let me put those away. So with those, I'm not so much concerned with those slides because they are not so problematic. Yeah, but the one with the bacteria, I usually give special treatment. Usually I drop over some alcohol before I wash it, or use some detergent or some very hot water. Yeah. So what I've got here is this is at least this is the fur of a sheep. I got this because we have got this wool blanket, and this is from a dog. Yeah, not a pet dog, not mine, but the one from my mother. And I would like to put those also under the microscope. And here we have the following issue: that hair generally, of course, is there to protect the animal from water, right? So it's greasy. So if I were to add water now on top, it's probably not going to, it's going to make a lot of air bubbles because the greasy fur and the water, they do not like to mix. So the best idea, I mean, I can try to show you, but the best idea in this case would be to use either oil, which I don't have, but I'm going to make I'm going to use U-Pearl, a mounting medium. And somewhere I do have tweezers. Okay, because simply it's easier like this, and so this, and let's have a look. This was from a wool blanket, and let's add a little bit of water here. Okay, and a cover glass, and let's see how this actually works. Okay. But this seems to be fairly highly processed wool. Yeah, maybe, And I need to, have a look here. And this is how it looks like, the wool. Okay. And let's go up a little bit. And I just want to show this to you for one reason as well, because especially at higher magnification, we're able to see something which is typical, which kind of gives it away that we're looking at here, the natural and not synthetic. Because if you look at the surface, where's the arrow again here? If you look at the surface, you see that it kind of is irregular. It has these scales, right? And that is typical for hair. Synthetic fibers would look very different. They would be much more homogeneous, and smoother. So this is actually one indicator, and another one is, look at this. Some of them are thinner, others are thicker, right? So you already know just by looking at it that this is indeed, not artificial synthetic, but actually natural wool. Because hair, or the fur rather, right? So again, you might every now and then, quite often I get questions, where do all of the colors come from? I just want to say that my microscope has a certain polarization setup, which allows me to change the colors around, like this. On the bright field, regular bright field, it would look like this. Okay, so that would be regular bright field. I don't know, I've not tried 4 times. And let's see, this would be now dark field. Remember the 4 times. I cannot get proper dark field with the 10 times objective. See, but with the 4 times, I can get dark field. So, and now let's look at the dog. Okay, Yeah, as promised, today's live stream is a little bit random because going all over the place with different topics. And let's try to put a little bit of dog here now. Yeah. A small amount is enough. Okay. Actually, I'm kind of surprised it takes water better than I expected. So. And this is the dog fur. And here too, if we go up with the magnification, we're going to see that it is highly textured. Yeah. You know that I've been occasionally. I'm kind of interested also in finding microplastics and other contaminants. Yeah, in whatever I look at. And one way of actually distinguishing them is that contaminants, either artificial or natural, yeah, are often much thinner, yeah, and not as straight as here. Yeah, so all of those fibers that you occasionally see, yeah, are generally in many cases not because of animal fur, but could have other origins. Yeah. Is human hair textured? Let's find out. Let's find. My hair might not be the best one to observe because I've got generally very dark hair, black hair. But as I'm not the youngest one anymore, my hair starts to become gray. So for this reason, let's take a sample from my hair and put it under the microscope and let's compare it. And indeed, the hair of mammals, and humans are mammals, indeed is to a certain extent similar, right? So let's do the following. Let's take a little sample here. Let's put it on here, and I hope that some of the, yeah, yes, I got some white here as well. That's good. Yes, that's too much. I think there is some white here as well. And the question is it also textured? And actually, maybe I should rename the. Maybe I should rename the title of this this live stream a little bit. So, if we're lucky, we're able to see now. So that is now my hair. And I don't know if I'm able to find. Let's go down a little bit with the magnification of where I have to look. Here's a white hair. Here, between the air bubbles. Okay. And the white one is more easily visible. Yeah, and you can see that. Not so much visible here, but you can sometimes see the central canal as well. Here it's better with a dark one, indeed. You have to really go up with the light intensity, and you're able to see that the hair actually has also some kind of something in the center here. Again, my hair here is relatively dark, therefore difficult to see. This one is a little bit brighter. Yeah, but you see also that there is, it's quite, also irregularly pigmented. Yeah. Do we use a polarization condenser? Yes. Not now. This one not. Okay, now I moved it away. Yeah. So this is now the natural. Yeah. It's basically not the natural color. Yeah, and here, yeah, it's when you shave yourself, yeah, and you put that under the microscope, then you can actually see some of the shavings, when they're on the side, and you can actually see the cross section of the hair as well. So it's also quite interesting because then you're actually able to see the central part that it actually has a different color. Yeah. So this is, yeah, basically, you see it kind of, that's another thing. If it is in contact with water, it will expand. Yeah, so that's basically what's happening right now. It will absorb the water and it will start to, expand a little bit. By the way, you can use this also as a way to, measure moisture. It's kind of interesting. Not all of those, not all of them have this central canal here for whatever reason. It's kind of interesting. And of course, different colored here. Yeah. So, okay. What time is it? Oh, it's already 1 hour and 20 minutes. I think I'm slowly going to call it quits. I hope I did not. I hope I didn't. But you can use them as a paintbrush. Actually, when shaving, you have those brushes as well. Yeah, it's camel hair normally. It would also be interesting to see how similar or different that is. Yeah. How do hairs grow and are the scale? The hair the hairs are formed. Hair is protein, 100% protein. The name of the protein is called keratin. That's just not carotene when in the carrots, but keratin. That's the name of the protein and is made in so-called hair follicles. The hair follicles are those cells that actually will make the hair protein. And those hair follicles are quite oily and greasy because they're oil glands. So that basically, as the hair grows out, that's why there's a thin layer of fat and grease on it. And this oil, of course, is also a very good food source, not only for bacteria, but also for so-called face mites. And some of you are probably gonna have already watched some of the videos that I made about those face mites, which live in the hair follicles, in those parts of the skin where the hair is actually made. So this is, So these are basically, the hair is not alive. It's not made of cells, but just like the fingernails, for example. But the cells will actually produce the protein, which then is deposited to make the hair. Yeah. So a hair of protein. So does this mean there is not from it? That's correct. Well, the cells make the protein, but they themselves are not made of cells. Okay. So it's kind of the product that of cells. So per definition, therefore, a hair is not alive. Yeah. So that is the thing. It's just so to say released by released is also not the correct term, but produced by the cells in the hair follicles. Yeah? So this is the reason why when you look at hair like this, you're not able to find cells because it's not cells. Right? It's just long protein fibers. Can I eat hair for protein? You shouldn't because it's actually quite dangerous. Because your stomach is able to digest it, of course. It's very acidic. It's not easy to be digested here because it's very strong protein, but the acid will actually break down the protein as well. It's not a lot of protein that you get. You'd be better off eating a little bit of meat or egg or fish or soybeans. Yeah, I'm quite sure that you're able to get more proteins from that. Yeah, so this is why it's dangerous to eat hair is because when the hair actually goes down your throat, it's gonna make you throw up. Yeah, because you cannot swallow it. Yeah, so this is actually quite dangerous. What I heard, right? Especially if you swallow it and then you have got hair hanging all the way down into your. No, don't want to think about it. Shouldn't be eating hair. Yeah, so this is the thing, and yeah. Let's put it this way: there are better sources for protein, and probably also have more higher quantity. Yeah, but indeed, at the university, long time ago, in chemistry class, what we've done at the universities is we took our own hair, and we had to digest it in hydrochloric acid, which is, by the way, also an acid in our stomach. We actually broke it down in a test tube, and then we analyzed which amino acids are present in the hair. Because protein is made of amino acids, so it's possible to do a chemical analysis of the hair. Right? So it was an interesting lab, but yeah. I digress. I digress. What I'm going to do now is I'm just going to call it quits for today. Okay. Yeah. Hope it was interesting. I hope I'm able to get it somehow fixed, the whole thing with the camera. But I don't know why it froze. But at least, yeah, at least we had an improvised solution for the remaining live stream. Yeah. Yeah. So yeah, okay. I'm going to leave it at that. I wish you a nice week. Happy microbe hunting as always. Yeah. If do watch also the other videos of this channel if you're interested in microscopy. Invite you to do so. And bye bye. See you again next week. All the best.